My name's Kelley Pratt. I'm a medullary cancer patient, and you might have seen me on our last session. I would like to, the session that we're in is titled CAR T-cell therapy for medullary thyroid cancer. We've got two excellent doctors presenting for us today. Dr. Donald Siegel is a professor of pathology and laboratory medicine at the University of Pennsylvania. He will speak first, and then we will have Dr. Roger Cohen, who's a professor of medicine also at the University of Pennsylvania. They're doing exciting new research, and I know that everybody is excited to hear about it. We've got 34 now, so I'm going to go ahead and turn it over to Dr. Siegel. Thank you for being here with us.
Great.
And we're glad to have you.
Thanks a lot, Kelley. So before I start my slides, I just wanted to introduce myself a little bit. So I'm a professor of pathology and lab medicine at the University of Pennsylvania. I've been here for about 40 years. Clinically, I run transfusion medicine, which comprises the blood bank, the apheresis unit, stem cell lab, and a laboratory that is called the GMP facility, which is a lab that creates novel types of cell therapies. I also have a research lab that I began about 30 years ago, where I create recombinant antibodies to various types of targets. And through much of my time here at Penn, those two kinds of activities, the clinical ones and the research ones with the antibodies, were quite distinct from each other. And because of the story that I'm going to tell you now, the two of them kind of came crashing together and led to what I'm going to be speaking about, which has to do with a potential new therapy, a cellular therapy for medullary thyroid cancer. So I'm going to start my slides. I hope everyone can see them. So I'm going to be talking about some research that we did here at Penn on medullary thyroid cancer. And then after I speak, one of my colleagues here, Roger Cohen, who's an oncologist, will be talking about a clinical trial that we opened here at Penn that utilizes what I'm about to talk to you about. So this is where I'm located right now. And up in the top are research buildings where I was doing my antibody stuff. And these are a couple of different hospitals here. And the clinical areas that I was just describing, where I do my clinical work. And I'm actually sitting over here where the star is, talking with you across the hall from this cell therapy manufacturing facility, and also my current research lab. And the way the story starts is back in 1991, when I came across this publication, which has a kind of a obscure title, Assembly of Combinatorial Antibody Libraries on Phage Surfaces, the Gene 3 site. Not a particularly exciting title, but what this was about was the beginning of the ability to create antibodies to virtually anything in a relatively short period of time. And you all know what antibodies are about and what they can do for you, especially with the COVID situation and getting vaccines, and everyone's talking about antibody testing and things like that. And even COVID antibodies have been made actually using this method that had been described back in 1991. And it turned out that, so this paper the first author was Carlos Barbas III, and here's his picture on the right-hand side. And he did this work when he was a postdoctoral fellow at Scripps Research Institute. And he grew to be a professor there. And he and I became friends when I talked to him about this publication, and he taught me about this method called phage display, which is a method for creating antibodies. And he and I became friends, and over a number of years, we actually taught a course on Long Island in New York at Cold Spring Harbor on how to do this. We would teach 16 students every year for about 20 years. And you can see down here at the bottom, he and I and a third fellow taught the course. And here we are. I'm over in the middle here. Here's Carlos. This is it's about 20 years ago. And we became quite good friends every year getting together. He lived in California. I lived, I'm here in Philadelphia. And then one year, in 2013, right before we were about to get together and teach the course, I got this email from him saying, I won't be able to attend the course this year. I just got the news of the discovery of several tumors in my neck area. Still don't know type yet. I hope I can call on you both for advice. And so, we were concerned about this. We didn't know what it was. I thought maybe he had lymphoma, something like that. In any case, he wound up having surgery out in San Diego. They had over 100 positive nodes in his neck area. And what it turned out to be was medullary thyroid cancer, which you all know about. And just to review, this is a microscopic image of a thyroid gland. And the cells that are lining these big open areas are the cells that make thyroid hormone. But then buried in between those follicles and those other cells are these cells called parafollicular cells, which make calcitonin. And because they make calcitonin, they're also called C cells. And it's interesting because it's not in every animal that parafollicular cells, the cells that make calcitonin, are actually in the thyroid gland. In other animals, they migrate down during development from the brain, and they actually are in other organs of the body, not always the thyroid gland. But in humans, they're in the thyroid gland. So when they become cancerous, they call it thyroid cancer. But as you all know, as well or not better than I do, that these are very distinct kinds of cells and distinct kind of cancer from the more common papillary or follicular type of thyroid cancer. And as you know, a lot of what drives the malignancy of these cells are mutations in a protein called RET. This is summarizing various places where there can be mutations. And just for interest, Carlos had a not very common mutation called the delta 632-633 mutation, which wasn't a mutation in a particular amino acid. It was actually the loss of two amino acids, number 632 and 633. And because of that, for reasons I won't get into, it actually caused two RET molecules to always be together next to each other, connected physically to each other. So it was a very bad mutation because these cells were continually activated and firing because of this particular mutation. So as scientists and clinicians here at Penn, thinking about something called CAR T cells, and I'll explain what that is in a little bit, chimeric antigen receptor T cells, and we had just developed these cells for treating lymphoma and leukemia, we wondered, could we make a CAR T therapy for Carlos for his medullary thyroid cancer? So the first thing that you need to do is identify a target on the surface of the C cells. And the target could be something very specific to what Carlos's example of medullary thyroid cancer, or it could be a target, would be even better if it was a target that would be on everybody's medullary thyroid cancer. Or actually, it could just be something that's on C cells. Not even whether they're malignant or not, because one of the things, as you can appreciate about thyroid cancer, is that the normal thyroid gland is one of the first things that is removed if you're diagnosed with this. So if you're designing a therapy that kills the parafollicular cells, the C cells, it would be okay if the therapy actually would kill a normal one too, because you don't have any normal ones. So that means that the bar is set a bit low for trying to find a target. In other words, if you were trying to cure brain cancer, you need a brain cancer specific target. You can't just have a brain specific target because you need the rest of your brain. But in this case, you don't even have the rest of your thyroid gland. And even if you did, it turns out we don't need C cells. When you have your thyroid removed, you take thyroid hormone replacement therapy every day, but you don't take calcitonin pills. So we were basically trying to find a target that was on C cells, that was also on medullary thyroid cancer cells. And the idea is if we could identify a target, we could make an antibody to the target using phage display, this method that Carlos had developed and had taught me. And then we could take that and make that into a CAR T cell. And I'll explain again a little bit what a CAR T cell is. And so what we did to try to identify potential targets was to analyze Carlos's tumor, and this long list of stuff you can't read, or just is a long list of different things that were expressed in his tumor. And we organized them by things that were expressed more in his tumor than in normal tissue. And right up at the top, you see calcitonin A, which makes sense that that would be highly specifically expressed by his tumor rather than normal tissue in your body, because that's what these cells make. But that's not something you could target normally. It's not on the outside of the cell. But then the second thing on the list here was something we had no idea what it was, GFRA4. And it had an R in it, and we thought that maybe stood for receptor. And receptors are things that are on the outside of cells. And so we thought, well, maybe GFR alpha 4 is a target that's on the outside of C cells and medullary thyroid cancer cells, and maybe not on the outside of other cells in your body. And we could use that as a target. So we looked in the literature, what is GFR alpha 4? And it turns out that GFR alpha 4 plays a very important role. It's this red thing here, a very important role in pulling together two red molecules, which are the blue things, when your C cells are activated. And so there's a protein called percephin, which is this green stuff, which binds to GFR alpha 4, which then binds to the blue, to red, and pulls the two molecules together. And so that's what GFR alpha 4 was. And so we said, is it specific to C cells? Or is it on every other tissue of your body? Because that wouldn't be such a great target. Already we didn't think so, because that's why it came up right at the top of the list in Carlos's analysis of his tumor. And so we looked in literature, and there was a paper actually from 2001 where they looked for GFR alpha 4 in all these different tissues, from brain to thymus, salivary gland, colon, pancreas, and so forth and so on, liver. And the only place they could find it was in thyroid gland. So that was very encouraging, because it suggested that really it's something that's quite specific to thyroid, because of the fact that in the thyroid gland are these C cells. We did some studies on our own, which, so this is a normal thyroid, and this little brown specks that you see here is where we were detecting GFR alpha 4. And then MTC1 is actually a piece of Carlos's tumor, and you can see it's filled with the GFR alpha 4. And then MTC2 is another example of medullary thyroid cancer that also has staining for GFR alpha 4. And then TT cells are actually a cell line that you can buy that was made a long time ago from a patient who had medullary thyroid cancer. And so these are cells you can grow in a dish. They're not pieces of tissue. And so we grew up some of these cells, and we centrifuged them into a pellet, and then pretended it was a piece of tissue and stained it for GFR alpha 4. And what you can see here is that it's chock full of GFR alpha 4. We also looked in other tissues for GFR alpha 4, and we found it in medullary thyroid cancer, in the TT cell line, and in thyroid gland itself, but we didn't find it, it's all negative, we didn't find it in any of these other tissues, which was consistent with what this Lindahl had published a number of years earlier. So we had a potential target, and now what we need to do is make an antibody against the target. And so using methods for making antibodies that Carlos had taught me years earlier, I made an antibody against GFR alpha 4, and the name of the antibody, the arbitrary name of it is called P410. And so the next thing that, once you have an antibody to a target, if you want to make what's called a CAR T cell, which is a cell that will use that antibody to go into your body and find the cancer target and kill it, you need to take the antibody and make a CAR T cell. So let me explain what a CAR T cell is. So CAR T stands for chimeric antigen receptor T cell. So a chimera in Greek mythology, and I'm sure many of you know this, It's an animal who had the head of a lion, the body of a goat, and the tail of a snake. And so why that relates to this biological thing I'm about to tell you is, here's the an antibody. In this case, it's an anti-GFR alpha 4 antibody that I just was talking about. And antibodies are, you may not know what they look like, these Y-shaped molecules, but you do know that they're proteins in your blood that are very specific for their targets. They're things you can make in the lab, like I made anti-GFR alpha 4. They're things that your body makes in response to vaccines, like the COVID vaccine. And the important part of an antibody is that at the tip here, VLVH, which I won't go into what that means, but that part of the protein is what is very specific for a target. And so we can easily make antibodies that are specific for targets. The problem is that just infusing antibodies into people, let's say people with medullary thyroid cancer, the antibody itself has limited ability to do something other than just bind to the target. It has limited ability to kill the target. Now, there are ways of getting antibodies to do that, but it's a little more complicated. Now, on the other hand, your body is filled with cells called T cells. T cells recognize targets also. And the thing about a T cell is it's a cell, it's alive. It can actually kill other things. And the way it recognizes its target is by this thing here called a T cell receptor. But it's very difficult from scratch to design a T cell receptor to a particular target. So what a chimeric antigen receptor is, it's a synthetic molecule we make where we combine the recognition portion of an antibody, something you can easily make in the lab, and we combine it with the inside part of the T cell receptor complex to create from a patient's T cells, cells that have this in it. And so that when you put these cells back into the patient, the antibody portion of this will recognize the target and will fool the T cell into triggering and firing and killing what this antibody is binding to, thinking that the thing that was triggering it was the normal T cell receptor here, which it isn't. It's been replaced with an antibody. So that's what a chimeric antigen receptor, or T cell, is. And so it's the head of an antibody, a body of a protein, and the tail of this receptor. So that's why it's called a chimeric antigen receptor. Okay. So so the idea is that we want to target medullary thyroid cancer with a GFR alpha 4 directed CAR T. And so here's a T cell that we would take out of a patient. It has its normal T cell receptor on here, which is not actually going to be playing any role in anything. And here is your C cell, your medullary thyroid cancer cell that has GFR alpha 4 on it. And so in the laboratory, after we collect your T cells using a process called apheresis, we use something called a lentiviral vector to put the gene into the cell that will teach the cell how to make the chimeric antigen receptor. The cell does that and puts the chimeric antigen receptor onto the surface of the T cell. When you put the T cells back into the patient, the hope is that the CAR finds the GFR on the surface of the medullary thyroid cancer cell. The T cell then secretes these toxins that go into the membrane of the membrane of the MTC cell and kills the cell. Now, I should say a little bit more detail about how this is made. So basically, patients who get CAR T cells, and CAR T cells are now, there's 6 of them that have been approved by the FDA. A few of them for leukemia and lymphoma, and the other ones are approved for myeloma. And they don't target GFR alpha 4, they target things that are on leukemia cells or myeloma cells. But the way the cells are made is all this basically the same process. So a patient will come to an apheresis unit, which is a place that has machines very similar to what the Red Cross uses if any of you have donated platelets at the Red Cross. You sit in a chair. I'll show you a picture in a minute. Actually, not even in a minute. So you sit in a chair, and your blood comes out of your body, goes through the machine. Everything goes back into your body through another IV. But what is extracted out of the blood are your T cells. And so your T cells then are in a bag, which then here at Penn go across the street to across the hall from where I'm sitting into this laboratory here, which does a whole bunch of things under very, very sterile conditions like that. That's what it looks like inside. And so what happens is the bag goes through a machine that purifies the cells a little bit more. The cells go into a process where a virus is added. That's the virus that puts the chimeric antigen receptor into the T cell. Magnetic beads with things on the surface are added to stimulate the cells to grow. The cells are put into bags that let the cells grow over a period of about 7 days. Then the cells are harvested and quality control steps are performed. An electromagnet is used to remove the beads. And then it's formulated into a final drug. And then the patient comes back, and generally in an outpatient setting, sometimes in a hospital setting, the patient will get the cells reinfused. And the hope is that the cells then circulate through the body. The chimeric antigen receptor on the surface will interact with any cells that have the target on it. In this case, it would be GFR alpha 4, and then it would kill those cells. Now, how did this start? So this started here at Penn a little over 10 years ago. This was the first publication that described the use of CAR T cells for treating chronic lymphocytic leukemia. And here's a video that was made, at the time. So these green things are leukemia cells, and this is a CAR T cell that's directed against something called CD19, not GFR alpha 4, CD19, because that's what CD19 is on leukemia cells. And you can see what's happening now is the CAR T cell just recognized the CD19, and it's just killing the CAR T cell. And so when this is actually used in vivo in a person, each CAR T cell can kill up to 10,000 other 10,000 cancer cells. And since this is a drug that's actually alive, these CAR T cells divide, make more of themselves, and actually hang around your body, almost like a vaccine in case the cancer were to come back. And so this was the first patient who was described in that paper I just showed you before. Patient number 1 of CAR T, it's called CART19 because the target was CD19. And just as an example, in July of 2010, his bone marrow was filled with these purple things, which were leukemia cells. And then a little over a month later, 7.5 pounds of tumor actually were removed from his, were killed by the CAR T cells, and then he had a normal bone marrow after that. He was the second patient. And recently, February of this year, we published a paper that talked about this gentleman and the previous patient, how what has happened over the last 10 years, that the CAR T cells are still detectable in their bodies after 10 years. What garnered a lot of attention with CAR T was this child, Emily Whitehead, who had acute lymphocytic or lymphoblastic leukemia. And she was the first child treated with these CAR T cells for leukemia. And she did very well. And would visit us afterwards. And then an international study was done for this indication for CAR T cells carried out at all these different hospitals. She testified with her father before the FDA as living proof of the utility of this type of therapy. The FDA approved this first gene therapy in the United States, which was this CAR T therapy for leukemia and then lymphoma. We had a little bit of a gathering in the building here to celebrate. And here she is this past May, 10 years later. And in fact, a movie was shown at the Tribeca Film Festival this past June about this whole story, a 90-minute movie about how she had failed, had gotten leukemia when she was 5, had 3 failed attempts at using conventional therapies, and how the CAR T cells actually did an incredible job for her. And so what this led to was the idea of trying to adapt CAR T cells for all different kinds of cancers. And the basic difference is the CAR T, what the target is. So like for her and for those gentlemen, the target was CD19, which is also the target for lymphoma. And here are just other organs that all can be cancerous. And then listed under here are all these different targets that people are going after to try to see if they could treat those kinds of cancers with CAR T cells. And so in this case, what we're trying to do here is treat medullary thyroid cancer with GFR alpha 4. So getting back to Carlos's story, we did research on what I just described, and it was published last year. And there's a lot of experiments, a lot of stuff in there. But just to show you just two pieces of data, this is a graph showing that as you increase the amount of CAR T cells to the amount of medullary thyroid cancer cells, this is done in vitro in a dish basically. You get the percent of cells that are killed goes up. So what this is saying is that these GFR alpha 4 targeted CAR T cells that we made will recognize GFR alpha 4 on a medullary thyroid cancer cell and kill the cell. Now, this is just the beginning. You really want to do it in animal models, but at this point, we were hoping we could quickly get something approved to be able to use for my friend Carlos, when unfortunately he passed away. He had a very aggressive form of medullary thyroid cancer. He passed away 6 months after he was diagnosed. He had a calcitonin doubling time of 2 weeks because of that mutation that he had. And so this was very, very upsetting. However, this is his family that he left behind, and his wife donated money to a group called REACT, and which I believe is now part of ThyCa. And with that funding, we continued the research, and we did studies in mouse models of medullary thyroid cancer, where we took mice and we put in those TT cells, those medullary thyroid cancer cells. And then about 9 days later, put in the CAR T cells. And what this is showing is that inside the mouse, the CAR T cells were killing the medullary thyroid cancer cells. These are individual mice, and this is averages of tumor volume, averaging the results of all these different mice. And then if you look at a different way of looking at it is here we have control mice where we put tumors in, and if you don't treat them with anything, the tumors grow. But if you take mice and put the tumors in and then give them the CAR T cells, the tumors don't grow. And this is where we take leukemia cells, which are called NOM6 cells, and we actually cause them to also express GFR alpha 4. And we do that so that we can put these cells into a mouse and treat it with the leukemia treatment and show that that works, because that should work, because these are leukemia cells. But those same cells that have had our target in it here also will, in exactly the same way, get treated with the GFR alpha 4 cells. So it's just another experiment. There are many other experiments that were done. That's all I'm going to show for that here. But we named this project chimeric antigen receptor lymphocytes with oncolytic specificity after Carlos. This is the Carlos project. And what the next step was to put all this data together with lots of other stuff I haven't talked about, and you apply for permission from the FDA to conduct a clinical trial. It's called Investigational New Drug, IND, which was granted, and then a clinical trial opened up, and that is what the next speaker is going to talk about, Roger Cohen. So let me end my slides here and stop sharing my screen. And I could answer questions now, Or if Roger is here, he can put his slides up. And here he is. Okay, great. Roger, is your sound on? Roger?
Good afternoon, everyone, and thank you very much for giving Dr. Siegel and me the opportunity to talk about this very interesting project, which has a very interesting backstory, but is also a very important project for what we refer to as solid tumor medical oncology. I'm a solid tumor medical oncologist. I take care of patients with lung cancer, head and neck cancer, and thyroid cancer, each of which represents a profound unmet medical need with decent therapies, but therapies that could still use improvement. And so I'm just going to take you through, if I can get my slides to work, the summary of this trial, telling you why we are conducting it, how we are conducting it, and who is eligible for the trial. It's a first-in-human trial, so these CAR T cells have not been given to people before. Therefore, it's important to evaluate the safety. It's also important to evaluate the feasibility of making the cells, Dr. Siegel having outlined to you a somewhat complex procedure in the laboratory for harvesting the cells, changing their genetic makeup, growing them up, making sure that they're not contaminated, and then being able to reinfuse them into people. And of course, with any of these clinical trials, we are keenly interested in any evidence of preliminary efficacy, whether that be on tumor markers or actual measurable tumors themselves. We also have some scientific objectives that are quite important. Just as was described to you with the CD19 CAR T cells, we want to be able to detect, find these cells in the blood. We would very much like to know if these CAR T cells are smart enough to actually get into the tumors. And we do that by means of a biopsy to see if these novel T cells have been able to migrate into the tumor, which is what has to occur in order for them to kill cancer. And this is an immune therapy project, so we want to look at a variety of immune and inflammatory markers in the blood, both before and after the CAR T infusions, because we are in part activating the immune system to attack cancer. These are the eligibility criteria for the protocol. In brief, adult patients with medullary thyroid cancer that either have never received because they don't want it, or have received and are no longer responding to a so-called multi-kinase inhibitor. And there are several that are FDA approved for medullary thyroid cancer drugs such as cabozantinib, selpercatinib, and pralsetinib. I want to point out in the eligibility criteria that we are completely silent on RET mutation status, because this particular product, this drug, this CAR T product, has nothing to do with RET mutations. It should work, if it's going to work, in patients bearing or not bearing a RET mutation. Patients need to have adequate organ function, kidney, liver, heart, lung, a reasonable performance status, meaning that people are ambulatory and capable of taking care of themselves in their home. They should have recovered from prior therapy side effects. They need something that we can follow, what we refer to as evaluable disease, which means something that we can measure or assess on a scan to see if the drug is actually doing anything useful. And of course, we want subjects of reproductive potential to use acceptable birth control methods, because we don't know what the effects of these CAR T cells are on developing fetuses. Exclusions are not many, but patients with an active infection should not be enrolled in this trial because we don't know if the CAR T cells will exacerbate or aggravate that infection. We are also excluding people with advanced heart disease, including abnormal heart rhythms that cannot be controlled medically. We don't want patients who are on high-dose steroids because high-dose steroids can actually damage the T cells that we're infusing. And so we're kind of working against each other if we're also giving patients high-dose steroids while getting CAR T cells. This is an immune therapy, and we are excluding patients with active autoimmune diseases requiring ongoing immunosuppressive therapy. So that would be, for example, somebody with lupus who is requiring active, powerful immunosuppressants to control their disease. And we are also excluding patients with central nervous system or brain metastatic disease from medullary thyroid cancer. As stated, this is a phase 1 trial that's looking at safety and feasibility of these CAR T cells following, and this is important, a step called lymphodepletion with traditional chemotherapy drugs, fludarabine and cyclophosphamide, drugs that will have no effect whatsoever on the medullary thyroid cancer, but do clear out some space in the blood and the bone marrow in order for the infused T cells to proliferate and we hope thrive. We are giving patients a single fixed dose of CAR T cells. The dose levels are listed here. There are 3 cohorts, and they will contain somewhere between 3 and 6 patients each. It's important to point out that the patients that were shown to you moments ago by Dr. Siegel also received a single dose of CAR T cells. So this is intended to be a single therapy that we hope will have lasting effects. The dose levels that are listed above are analogous, similar to those used by our institution and others in studies evaluating the safety of other CAR T cells in other so-called solid tumors, such as mesothelioma, ovarian cancer, etc. The lymphodepleting chemotherapy with fludarabine and cyclophosphamide is given over 3 days, and then that is followed by the infusion of the CAR T cells. These are the doses of fludarabine and cyclophosphamide. They are very standard by now in this field. Patients are given a single fixed dose, as stated earlier, and the study itself will consist of 5 sequential phases, including a screening phase when we meet the patient, tell them about the trial, obtain written informed consent. Then we proceed to harvest from the patient white blood cells, T cells, and then manufacture the CAR T product in the laboratory. The treatment phase, which I summarized on the preceding 3 slides, is the lymphodepleting chemotherapy and then the infusion of the CAR T cells. And then there's both early as well as long-term follow-up, the latter for up to 15 years post-infusion. So we will look at the patients, examine them, talk to them weekly for 4 weeks after the initial infusion. We will assess them monthly for 6 months and then every 2 months until month 12. And as stated above, effectively are committing to following our patients' lifetime for at least 15 years. I'm not expecting you to read this, but this is just a graphic of the process that I outlined in the preceding slide. There is a retreatment provision, so there probably will be leftover CAR T cells in a freezer, and we do have the ability to give a second dose of CAR T cells at the discretion of the physician investigator, and that of course depends on whether the patient tolerated the initial infusion reasonably well, and of course whether there's a perceived need for a second infusion of CAR T cells. Potential side effects are something that you always have to be concerned about with a new therapy. And although we have done our very best to minimize the chances of such side effects occurring, they nevertheless will be looked at and looked for because they have been an issue with some of the other CAR T products, including the CART19 product that is associated with profound anti-cancer effects in patients. I think the biggest concern we have is that we could inadvertently, unintentionally target and damage a normal organ in addition to medullary thyroid cancer cells. And the purpose of this slide is to remind you of something that Dr. Siegel told you a few minutes ago, which is that when we look at human tissue specimens in a bank, or we look at primate specimens, you will notice that all of the boxes in this diagram, with the exception of medullary thyroid cancer and thyroid cancer cell lines, are negative. So there was no reactivity of the CAR T cells with other human tissues. But our tests are not perfect, We hope that we have an accurate picture of what could happen, but we will be looking for what is referred to as off-target toxicity, inadvertent targeting of another human organ. This is an immune therapy. We are infusing immune cells, T cells, into people, large numbers of them. Infusion reactions are possible, including fever, chills, nausea. We will pre-medicate for these with medications such as acetaminophen or Tylenol, ibuprofen, Advil, and we will continue to administer these medications if fevers develop. We will intentionally not use drugs like prednisone unless we need to. Prednisone is a corticosteroid and could actually work against the CAR T cells, but of course, if a patient has a life-threatening side effect, we will be forced to use them. If there's fever, we will always think about the possibility of infection and treat with antibiotics if they're indicated, as well as other measures, including fluids. If low blood pressure occurs, we have medications that can raise the blood pressure, and we will do cultures and other appropriate medical management for fever and the possibility of infection. Rashes do occur with immune therapy, and if they do occur, we will consult dermatology, probably do a biopsy, and treat as appropriate. Cytokine release syndrome is a very special potential toxicity or side effect of CAR T therapy, which includes a syndrome that may include high fevers, chills, rashes, muscle and joint aches. I won't read all of these particular side effects to you. Mild cytokine release syndrome, which occurs as a result of the intended effect of the CAR T cells. We want them to do something. We want them to become active and quickly kill cancer cells. Cytokine release syndrome is too much of a good thing. Mild cytokine release syndrome can be managed with acetaminophen, but if the cytokine release syndrome is higher grade, meaning that it's more serious, we may, in fact, need to treat with steroids to prevent further activation of the immune system, antihistamines, drugs such as Benadryl, and other supportive care measures. The University of Pennsylvania has been in the forefront of helping to develop and use anti-cytokine drugs such as tocilizumab or siltuximab, again, to specifically turn off an out-of-control immune response. And again, the purpose here is not to read this to you or even have you look at it, but this is a published guidance for the management of cytokine release syndrome that was developed almost 10 years ago now by investigators at the University of Pennsylvania treating patients like Emily Whitehead with the initial first-generation CAR T cells, during which we figured out a lot of the management paradigms for dealing with side effects. Neurological toxicity is another potential side effect that can occur. How it occurs, why it occurs is uncertain. It may even occur after the CAR T cells are given, after cytokine release syndrome. Thankfully, it's usually self-limited, but there can be brain swelling, which we would of course have to treat with medications and consult neurology. And finally, this is my last slide, there are some theoretical possibilities that we will be monitoring for in the blood, including the generation of what's referred to as replication competent lentivirus, which means that there is now a virus in the patient that has occurred as a result of infusing the CAR T cells. It hasn't happened, but we need to look for it. The T cells could proliferate or divide, multiply out of control, and that would be treated with prednisone. And finally, with any genetically engineered product, and even though this has not occurred, you always have to worry about the possibility that there could be genetic changes in the DNA of the patient that could even cause new cancers. Fortunately, this has not occurred, and there are a lot of tests that we will be doing on these patients, as well as patients in other studies, just making sure that this does not happen. So that concludes the slide presentation that I had, and Dr. Siegel and I are available for questions either in the chat or from the audience live, if you will. And so I'll turn it back to the moderator. Thank you.
Thank you so much, Dr. Siegel. I do have two questions. This is Kelley. The first question is, I'm not sure if you can see these questions.
Yeah, can I, I'm trying to turn my video back on if possible. If they could, it says that the host has stopped my video, if they could.
Yeah.
Yes, I'll ask him.
The question's in the chat, I can see it.
Yeah. Which question?
Okay, well, I've got a couple in the Q&A also. The first one is, are CAR T cells treatment used on other types of cancer besides medullary? I know you answered, but if you could just touch on that.
So this study will be the first one to use them on medullary thyroid cancer, but they were developed initially for treating leukemia and lymphoma. And there are 3 FDA-approved CAR T therapies for those diseases. There's also FDA approved for myeloma, and there are dozens of other CAR T clinical trials going on for virtually every organ of your body, from glioblastoma in your brain to ovarian cancer, pancreatic cancer, you name it. And so this is an incredibly active area of research using cellular therapies for treating cancer.
Thank you. I've got another one. I'm so sorry. Has any other treatment ever targeted GFR alpha 4 in humans? And have there been any participants in the trial yet?
So I can answer the first question, which would be that no one's ever targeted GFR alpha 4 before, because the only reason they would, from what we can tell, is for treating medullary thyroid cancer. So if it had been targeted before, then someone else would have done this already. I can let Dr. Cohen, who's in charge of the trial, answer the second part of that.
Yes, so the GFR alpha 4 target is very specific for medullary thyroid cancer, which is why these cells are not likely to be used for any other type of human cancer. That is one of the most important, unique, and special features of this particular project. We are about to treat our first patient. And so we are very grateful to the generosity of patients being screened for this trial, and we are about to treat our first patient. The agency will only let us treat 1 patient a month, so we do have another patient lined up as a follow-up to our first patient, depending on how it goes.
Thank you. I've got another question. How long do study participants need to be off targeted therapies before treatment with CAR T?
So we have recently changed our requirements. And so it's not very long. It's approximately 2 weeks. It's expressed as the 5 half-lives of the drug. A half-life of the drug is the amount of time that it takes for the level of cabozantinib, for example, to drop to 50%, and then 25%, and then 12.5%, and so on. So we have deliberately tried to make the period that you're off targeted therapy as short as possible, recognizing that many patients need to be on the targeted therapy for as long as possible. We don't want the targeted therapy and the CAR T therapy to overlap, but we have, in cooperation with the FDA, minimized the time off targeted therapy.
Great. I also have one. Landon is asking, what about other types of thyroid cancer?
I'm going to let Dr. Siegel answer that question because he's very interested in that particular area.
Yeah. Well, so I mean, again, the GFR alpha 4 targeted ones would not be of use for the other types of thyroid cancer, like papillary and follicular. But similar to the approach that we've used for the medullary, we're actively using that approach to try to identify unique targets on papillary cancer. That's the first step. And then after identifying targets and examining their potential presence elsewhere in the body and seeing if they're good targets, then we'll develop antibodies and CAR T cells and progress along the same lines that we've done here for the medullary.
Okay, thank you. Next question is one that's always on our minds. Robert asks, this being in the USA, what are the costs of these therapies, and does insurance or Medicare cover?
So I will answer that question. It is expected that insurance will or would be required to cover the cost of the therapy, which means scans, blood tests, doctor visits, and where there's to be a need for hospitalization, the cost of the hospitalization. The drug itself is very expensive and it's free because it's part of a clinical trial. The drug trial is being sponsored by philanthropy. It's not sponsored by the pharmaceutical industry. So there is a very limited budget in the trial, which is enough to cover the cost of manufacturing the drug and so forth. In the United States, with respect to private insurance or Medicare, there are a number of laws that have been written over the past 25 years mandating coverage of clinical trials that are FDA approved for patients who are in need of such trials. The issue is a little trickier with overseas patients because they don't have US health insurance, and it is always a struggle to figure out how to pay for those particular patients. We do have international patients who have expressed interest, and we have one in particular who's lining up funding in order to get treated on the study.
Thank you. I just got a couple more questions. What about anaplastic thyroid cancer? I think you kind of covered that, but I think they might have missed it.
Well, that usually has evolved from so-called differentiated thyroid cancer, which means cancers such as papillary. So that's in the papillary or more traditional thyroid cancer family. So that does not bear the target, GFR alpha 4, and therefore would not be suitable for treatment on this trial.
Perfect. And how long do you expect this will remain available to patients in a clinical trial phase? And thinking years or
It's going to depend on how rapidly patients are enrolled. I can't really treat more than one patient a month. And finding patients who are suitable has been a challenge because, in a way, fortunately, medullary thyroid cancer in a number of patients is a relatively indolent disease with prolonged periods of disease stability. And patients in that particular category may not be keen to be in a phase 1 clinical trial. Having said that, my guess is that the trial will be open for another year or two. And of course, if there are benefits seen in patients, the trial will end quickly. And if the drug results in severe side effects, the trial could also end quickly. My hope is that it will not have serious side effects and that it will have benefits and therefore would be open for another year or two.
That's awesome. I think I have one more. I think we have time for this one more. Cancer cells are good at evolving to escape targeted drugs. Is it even possible for MTC cells to evolve to not have the GFR alpha 4 receptor? That's a good question.
That's a great question, which I will defer to Dr. Siegel.
Yeah, so it's a great question, and we don't know the answer to it. We would hope that the cells are killed before they have a chance to continue to grow and lose it. But of course, this is something that we would look for.
Okay, and then I'm going to sneak another one in. Lots of questions. I like it. My husband's cancer has spread to the liver. Is it right to assume that this treatment would also target the cancer in his liver?
Yes, so it should target the cancer wherever it is located, meaning lymph nodes, lung, bone, liver. You may remember that we specifically excluded patients with central nervous system or brain involvement precisely because of concern that the CAR T cells may not actually be able to figure out how to get into the brain. But the liver, bone, lung, nodes, and so forth, the idea is that the CAR T cells will go everywhere where there is cancer and attack it in those locations.
Thank you. And another one, I missed the beginning, but how does this method deal with the PD-1, PD-L1 issue?
It doesn't. So this is not that kind of an immunotherapy. So those are the PD-1, PD-L1 monoclonal antibodies, drugs with names such as pembrolizumab, which are investigational for medullary thyroid cancer. drugs that are very active in melanoma, lung cancer, head and neck cancer to some extent, and are being evaluated in medullary as well as other thyroid cancers. But it's a completely different mechanism of action. In brief, what those drugs are doing is getting the patient's own T cells without being engineered to attack the cancer, effectively by unmasking the cancer so that the patient's immune system can now see it, recognize it, and kill it. Our therapy is quite different in that we're taking T cells out of the body, we are changing them genetically, and then reinfusing them. Should we be doing all of these therapies at the same time? That is a story for another day. You have to first start small with one immunotherapy at a time before you can begin to combine things.
Okay, guys. If there aren't any more questions, I think it looks like we're about out of time. I really want to thank Dr. Siegel, Dr. Cohen for your time today, taking the time for us. We're always looking for new therapies, and this is really exciting for us.
Thanks very much, Kelley.
Thank you.
Thank you, Kelley. Thank you to the organizers. Thank you to ThyCa for involving us and listening to us. We really appreciate it. It's an honor to speak to you today.
Yes.
We'll be excited to hear some results.
Okay.
Thank you so much.
Bye-bye.
Thank you.
Bye-bye.